So first thing first, Arabidopsis thaliana is a model plant. So this is an Arabidopsis thaliana. This plant belongs to family Brassicaceae. Which family?
Brassicaceae.
Brassicaceae family. So Brassicaceae family, you know, Brassica plant. There are Brassica napus, Brassica oleracea, like mustard, Brassica campestris, sarson, our local sarson, and then we can say many types of plants like that is used for cooking, and then you can say cabbage, you can say cauliflower. Brassicaceae family plants. So Arabidopsis thaliana is also a Brassicaceae family plant. Is a model plant. Why it is a model plant? Have a look here. Number one, this plant is very small in size. If you look at this plant, so this is the plant. It has a max height of 1 feet, 10 inches, 11 inches, 1 feet, maybe maximum 15 inches. So is small in size, so it is very easy to handle this plant. Why Arabidopsis thaliana is a model plant? Model plant means we do a lot of studies on this plant to understand different molecular mechanisms, genetics, functions, functional annotation of the genes. Like we can do tilling, we can do gene knockouts through CRISPR-Cas system, etc., and we can knock out some genes, and then we can look at what their functions in plants. So this is why Arabidopsis thaliana is a model plant, because we can study and understand different types of processes like germination, plant growth and development. So when this plant germinate, it makes a rosette of there. Its leaves looks like this, like rose petals. They are arranged in a pattern that you can say is like pattern. So afterwards, from these plants, from these leaves, a stem-like structure, you can say stem originates, and it has very small branches. And after some passage of time, it develops some sleeks. Sleeks, you can say pods. Pods like pods, you can say. Like in legumes, we have already studied that there are pods like uh, chickpea pods, and each pod has 2-3 seeds, right? Can you hear me?
Yes, sir.
Yeah,
Yes.
sorry.
So like chickpea, every pod has two to three seeds, but these pods, similarly, brassica plant has leeks. So there are multiple seeds inside the brassica. You can say it is not relevant to, but it is more relevant to brassica, sarson, etc., etc. So they have sleeks, and in sleeks there are seeds, like maybe 20, 30, 40, 50 for different varieties. For this plant, these leeks have at least like 80 to 100 seeds per leek. So one plant can produce. So if you have a look at this picture, you can see that this is the rosette leaves. Rosette means making a structure like a rose. And this is one leaf. These are multiple leaves. And after some time, there will be usually a single stem. So single stem, it can have some branches, leaves. So these rosette leaves are there, but on the branches, there are narrow leaves. And finally, there are sleeks. You can say this is a green sleek, and this is a mature sleek. When the sleek matures, or when its color fades and turns, it has maybe tens of seeds, 40, 50, 60, 20, maybe more than 100 in single sleek. Because in one sleek, there are many seeds. So from one plant, we get thousands of plants. So number one, this is a model plant because this plant is very small in size. It it is easy to handle. The first reason for this plant to be a model plant. The second reason for this plant to be a model plant is that this plant produces thousands of seeds. So this is the second reason of this plant, model plant, that this plant can produce thousands of seeds. So if you have thousands of seeds from one plant, you can use them to study multiple things. You have a lot of seeds, very good. Now you can study germination, you can study the structure, and you can use them for transformation, you can use for tilling, anything. So plant research, you have to grow the seeds, and for growing the seeds, what you have to do is need seeds. So because this plant can produce thousands of seeds from single plant, so it makes it easy to study different aspects of plants, genetics, genomics, etc., etc. Okay, the third reason. The third reason is the genome of this plant is very small. So number one, this is a small plant. It can be handled very easily. You can grow them anywhere, like even at very small place, in a small growth home, in a small growth chamber. It's very easy to grow. It is very, and you do not need a lot of fertilizer for these plants. Usually, if you have peat and mixture of soil, it is plants. So easy to handle. Number two, this plant produces thousands of seeds, so it makes it ideal for studying a lot of plant science. Number three, the genome size of this plant is very small. That is 114.5 million bases. 114.5 million bases. 114.5 million bases
सर स्नैप
only.
ऐड कीजिएगा.
It is very small genome size. This is a very small genome size. If you compare it to the other plants, for example, let's see wheat. You know wheat, right? So wheat has a genome size of gandum जिसे कहते हैं. It has a genome size of 17 gigabases. What is genome size of wheat?
17 gigabits.
17 gigabases. So it means that if you say giga to mega, uh, 17 gigabases. It means it is how much? 17,000 megabases. So if you say 114 megabases, it is only 0.114 gigabases, which means that if you multiply it with the 9, 100, 9, and are you understanding it?
Yes, sir.
Yes, sir.
If you multiply 114 mega to 9, it will be 1 gigabase. So 7 multiplied by 17, 159, 153, right? Yes. So it means that the genome bread wheat that we use for making bread in Pakistan and various parts of the world is 153 times bigger than Arabidopsis thaliana. 150 गुना बड़ा है उससे. तो इसका मतलब क्या हुआ कि 150 छोटा होने का मतलब क्या है Arabidopsis का जीनोम का, कि इसके अंदर जो जीन्स होंगी उनको स्टडी करना इजी होगा. नंबर ऑफ जीन्स, so the lesser number of the 150 times lesser size of Arabidopsis thaliana genome as compared to bread wheat means that this has less number of genes. Yes, but in bread wheat there are 1 lakh 25,000 genes, 0.1 genes. But in Arabidopsis there are almost 25,000 genes, 5 times less. So number 3, why Arabidopsis thaliana is a model plant? Its genome size is very small, 114.5 million megabases or million bases. Megabases, million similar. 10 raised 6. So wheat has 17 gigabases, which means that Arabidopsis thaliana genome is 153 times than bread wheat. So bread wheat has 125,000 genes. Banana has only 25,000 genes, which is 5 times lesser than the bread wheat, which means that studying the genes in Thaliana will be much easy. So why Arabidopsis thaliana is a model plant? Number 1, this plant is very small in size. It 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 can be grown anywhere. It can be handled easily. Number 2, plant produces thousands of seeds which can be used for further studies, and it makes the genetic studies plant very easy. Number 3, its genome size is very small, so it is very easy to understand how different genes are functioning, how different genes are working in Arabidopsis thaliana. And number 4, finally, this plant grows very quickly. This plant grows very quickly. How much fast? You can say this plant grows in 10 to 12 weeks. Maybe you can say sometimes 8 to 10 weeks. So from their germination, when you will sow these seeds, within 9, 10 weeks, maybe 10 weeks and maximum 12 weeks, means within 3 months, within 2.5 months, you will get the seeds. It means any study, even if it involves studying the seeds, it will be finished within 2.5 months. But on the other hand, if you look at wheat, rice, other plants, cotton, etc., etc., it will take at least like 20 or 21 weeks, maybe 25 weeks. Yes, 25 weeks. So it means that this plant takes almost half or sometimes even less time in maturity as compared to the other plants. So this is the fourth reason why this plant is being used as a model plant. And finally, fifth reason for this plant to be used as a model plant is that transformation protocols have been optimized for this plant. Because this plant is a model plant, hundreds of labs all over the world are working on this plant. And those hundreds of labs have successfully developed different transformation protocols like done using floral dip method, using agrobacterium mediated transformation, etc., etc., to develop transgenic plants. So that's why it is used in hundreds of, maybe thousands of labs all over the world for research purpose, basically. So it is used in the field of plant science, genetics, agronomy, evolution, germination, plant growth, plant development, gene expression, gene knockout, CRISPR studies, tilling, etc., etc. Tree knockout mutations, anything you can name it, each and everything has already been performed in this plant, and most of its genes have been already characterized or studied for its functions. That's why it is a model plant. So, in summary, number one, this plant is a because this is very small in size and it is easy to handle. Number two, this plant grows very quickly and produces thousands thousands of seeds from a single plant that can be used for further genetic studies. Number three, its genome size is 114 million base, which is very small as compared to, for example, wheat, which is 150 times bigger than this. And then this plant, what we can say is, grows very quickly and it gives seeds in 10 to 12 weeks, which is almost half of the time that is required by major plants to give the seeds. And finally, the transformation protocol for these plants have been that can be used for genetic studies. So number one, because we are talking about the seed culture, so many labs are using this method to culture the seeds. So how we can culture the seeds? Number one, because we will use MS media, and media, we already have studied this protocol that there are a lot of nutrients, everything is there. So first thing is we have to utilize the seeds. So number one, we last time discussed how to make 75 70. We will prepare 70% ethanol, and we will take a few seeds, maybe 50, 100, 200, 300, depending upon size of your experiment, and you will dip them in 1 ml of ethanol, and you will turn the Eppendorf tube for 1 minute. You will turn it, you will rotate it. So each and every exposure to 70% ethanol. Number two, there are incubator shakers. uh, Incubator shakers. So there are different types of incubator shakers. Because you are not in lab, so I will show you on internet. For example, this is one incubator shaker. So these are kind of incubator shakers. They are usually available in labs, and they are closed. You can open them. You can set the temperature. You can set the RPM rotation. So what you can do is you can put your up and off. So there are some small. These are the bigger places, but there are sockets as well where you can place your up and off tubes. So you will place your up and off tubes, and you will set the temperature to almost centigrade, 22 to 25 degrees centigrade, and you will set the shaking at 100 to 120 RPM. So this is an effective one. Maybe if you set it to 200, due to high speed, the seeds can be damaged. So usually you will let the seeds agitate for 15 to 18 minutes. So what you will do is you will take the seeds out, and you will take them with the hand so that the seeds are not stuck at one side of the up and off tube. Discard the ethanol. So this thing the two steps are done, can be done. Definitely you can all understand they can be done at in the lab environment, open environment. You are just ethanol, and ethanol will kill the pathogens, so no problem. You can do it in open lab, open space. And then, because now you have four and a half tubes, and you have placed them on the incubator shaker, fine, no problem. But now from the step 3, you shift to laminar flow hood, because if you do not, there will be contamination, right? So you will open the up and off tubes and discard the ethanol. And then what you usually do is you will put 1 ml of absolute ethanol, means pure ethanol, and you will wash it. What you will do is you will put ethanol, and for like 20-30 seconds, maybe 5 seconds, depending upon your confidence, you will what you will do is you will just wash it with absolute ethanol, and after that you will discard the ethanol, and you will repeat this process for 2 times. So why you are doing it, how you will do it? You will first, you will take out your incubator shaker, and after taking them out from incubator shaker, what you will do is you will place a tube. So all seeds will settle down. The seeds will settle down on the bottom of up and off tube. So by using ml, uh, you can say micro pipette, which uses blue tips, you will do the maximum amount of 70% ethanol and discard it. And using a fresh one, using a fresh blue tip micro pipette, add 1 ml of absolute ethanol, now absolute ethanol, and you will close the tube. You will shake it for 20-30 seconds, at least 10-15 seconds, and then you will place the tube again. The seeds will again shut down. You will discard. What you will do, you will discard the absolute ethanol, and then you will put ethanol again, shake it for 20-30 seconds, and again let the seed settle down on the bottom, and again remove maximum amount of the 70% ethanol. So 70% ethanol does not kill the microbes, does not kill the seeds. But if you leave your seeds in 70% ethanol for 2 hours or 3 hours, your seeds will die. But because now this is absolute ethanol, if a very small amount of it is there, no problem. What you will do is you will now place the seeds in laminar flow hood. You will place of tube open, lid is open for at least 3 hours or overnight. Overnight means for whole night. The ethanol will evaporate and seeds are dried. बात समझ में आ रही है कि अब चूंकि 70% एथेनॉल कर देता है सीड्स को, लेकिन 100% नहीं करता, थोड़ा बहुत अगर रह भी गया तो कोई इशू नहीं। लैमिनार फ्लोट को यूजुअली हम उसका क्या करते हैं, उसके अंदर फाल एपन ऑफ ट्यूब्स को रख देते हैं। स्टैंड होता है, आपको पता है, इनका छोटा सा स्टैंड के अंदर रख देते हैं, उनका लीड ओपन कर देते हैं। पहले जो है इसका शटर या किसका, लैमिनार फ्लो हुड का, और फिर उसका फैन बंद कर देते हैं। तो इस तरह से उसमें जो कंटेमिनेटेड आएगी उसको 3 आवर्स के लिए या पूरी रात के लिए छोड़ देते हैं आप। सारा जो एथेनॉल है वो उड़ जाएगा और आपके पास ड्राई सीड्स आ जाएंगे। सो व्हाट यू कैन डू इज you can use them in two ways. Number one, you can spray them on a medium, or you can use autoclaved sterile water, autoclaved distilled water. Like for example, take a pound of tube, the seeds are there, put 100 microliter or 50 of water, autoclaved distilled water, and you can pick using a pound of tube, using micro pipette. The micro pipette, what will happen? The water, your seeds will also come inside the blue tip. So from there you can drop them drop by drop the media. For example, you are using petri dishes, drop them there. So there is another situation. If you do not have a lot of time to wait, what you can do is in step 3, here in step 3, instead of absolute ethanol, you can use autoclaved distilled water. Autoclaved distilled water, and you can wash them 2 times with the autoclaved distilled water, so that 70% ethanol is completely removed. After doing it, you will directly move to, for example, step 6. Because now you do not need seeds to dry, because it was required to evaporate the ethanol, because it is not required, washed 2 times with autoclaved distilled water. So you can again put a few amount of 100 microliter of water and use it to disperse on media by using blue tip. Blue tip means 1 ml blue tip that is attached to 100 microliter micro pipette. Is that clear? So cautions: do not leave seeds in ethanol for too long, it may kill the seeds. Do the steps 3 and 6 in sterile hood, and the seeds are ready. But another thing is also needed. What is it? We have already studied that we have to make MS media. We have already studied how to make 1 liter of MS media. This is just a repetition. So here we are saying instead of 1 liter or 1000 ml, we are making 100 ml. How much? 100 ml. So this amount will be instead of 4.4 gram, it will be 0.4 gram. It will be mixed in distilled water, and it will be stirred. And then we will add, instead of 30, because it is 10 times lesser, we will use 3 gram of sugar. We will make the volume up to say 90 ml. We will use sodium hydroxide or hydrochloric acid. We will adjust the pH to 5.8. Then instead of 8 gram of agar, we use 0.8 gram of agar. and we will make the volume up to 100. And afterwards, we can also place it at agitator so that it is completely mixed, right? Agar is completely mixed. So after doing this, we will autoclave it for 15 to 20 minutes. After cooling, 25 ml of warm media, not the cold one, because it is all dry. 25 to 55 degrees Celsius is poured into patty dishes inside the laminar flow hood. Plates are left in laminar flow hood for 25 to 30 minutes so that the media solidified. We have already discussed. Seeds can be transferred to media plates by sprinkling. If the seeds are dry, we have already discussed. If we leave them in what we say is mm, overnight, the seeds will be dry. We can use sprinkling, spraying them in suspension of 100 to 200 microliter of sterile distilled water. We can sprinkle them, or in suspension, we can make a suspension in microliters of sterile water by using the sterile tips during the micropipettes. Plates will be transferred to grow chamber for germination of seeds and growth of plants in controlled condition like light temperature and humidity. We have already discussed in theory and practical class that the temperature should be like 25 to 25 degrees and 22 to 25 degrees centigrade. Light should be around 4,000 to 25,000 lux. Humidity should be like 90%. So seeds will germinate in 2 to 3 days, and you will get good size of plant in 2 to 3 weeks. So this will be your project as well. You will do this experiment because this is an online class. So I cannot show you how to do it. So in the next class, hopefully it will be physical from Wednesday. I am hearing that tomorrow the class is already online, but from the Wednesday it will be on campus. Hopefully in the next class I will show you how to do it, and then you will make your groups of 5 and you will finish this experiment, and it will be of 20 marks, and it will be like uploaded as one of your assignments to your what you can say is your portal. So this is all. Right now I will just stop this sharing, and I will also stop recording. I hope I have recorded. So stop recording. Yes. So